Enzymes are the biological catalysts which are capable of accelerating the rate of a chemical reaction. The functioning of a chemical reaction must occur in case of enzymes in a biological organism's body. In case of biological organism, the temperature, pH, the body fluids, everything is in a maintained condition. So for the activity of enzymes, everything has to be in a same controlled fashion. If there is any variation, the rate will be affected. So for the production of various enzymes, the use of microorganisms are done by the human beings to prepare large quantity of required enzymes with the help of uh, these micro industries, which can be any kind of microorganisms. Since they proliferate very fast, so the production can be done at a large scale with the help of bioreactors. So the first type of enzyme which is formed is amylases, and these amylases are formed from Aspergillus, Rhizopus, and Bacillus. These three, two of them, first two are fungus, whereas Bacillus is an bacterium. So three of them collectively help in the process of production of amylases. What is the function of amylases? If you all remember, amylases are present in saliva, ptyalin. It has presence of salivary amylase, which proceeds or which helps in primary digestion of carbohydrates in our mouth when we are making or churning the food to form food bolus. Other than that, amylases are also secreted by the gastric juices in order to facilitate the process of digestion of carbohydrates. So, conversion of carbohydrate to smaller particles, that is glucose, is done with the help of amylases. If you think about the uses of amylases, they are formed by degradation of starch. They are used or they can be formed from alcoholic beverages after the ethyl alcohol is formed. So after production of ethyl alcohol, it can be further hydrolyzed with the help of Aspergillus, Rhizopus, or Bacillus in order to form amylases. Textile industry requires the presence of amylases again for the production or for the breakdown of any carbohydrate if it is present in the jute or in cotton or in um, any substance which contains carbohydrate content or is obtained from plant products. Paper industry, because paper is used for production of paper, and or even the pulp of various wood or leaves or certain substances are used for production of paper. Over there will be presence of carbohydrate and in the form of starch, so that has to be converted at first into ethyl alcohol, and then further it can be converted to amylases. And in case of bakery industries, the confectionaries, also we require the presence of amylases, or we can extract the amylases also from the bakery industries. So because they all are using these three type of microorganisms for their functioning, and we can extract the amylases from them. Next is about the lipases. The function of lipases is to break down the oily substances or fats. So lipases are present in our stomach juice or gastric juice, I should say, for digestion of fatty acids, and with the help of changing the environmental condition by addition of bile, the pH, the entire system, it becomes basic and or alkaline, and because of the alkaline medium which is created by sodium bicarbonate, the action of lipases can be seen for complete breakdown of the fats into fatty free fatty acids with the help of lipases in the human beings. Similarly, the same lipases can be formed or can be extracted from Candida. Penicillium and Rhizopus. I'm underlining them because I forgot to underline them. And you should remember that how to write a scientific name. Three of them are fungus, and from them it can lipases can be extracted. These lipases are used for for production of detergent, because the main function of detergent is to clear the stains, right? If there is any stain of food or any oily substance, then that can be only cleared if there is a micelle formation taking place. That micelle formation can only occur with the help of lipases and water. So lipases are main ingredient for the laundry industry for removal of stains in clothes. Next kind of um, enzyme required is pectinase and proteases. Pectinase and protease. Let's discuss about each one of them. Pectinase, if you remember the outer covering of in biotechnology, we did study about certain enzymes which are capable of delaying the time of ripening in case ripening of tomatoes early. So over there, the pectinase degradation was discussed. What is the meaning of pectinase? The outer covering of any fruit or vegetable is made up of pectin, and um, it can be broken down with the help of pectinase. So for the breakdown of outer covering of fruits and vegetables, we require pectinase, and it can be obtained. That is the that is about pectinase. What about protease? Proteases are substances which are capable of breaking down proteins into smaller particles like the amino acids. So both these enzymes are extracted from Aspergillus niger and Bacillus. Aspergillus niger is a fungus, whereas Bacillus is a bacterium, and both can be used for various purposes. Like, first of all, pectinase is breaking down the food. So first is if you think about fruit juices or fermentation, like production of wine over there, whenever there is a fruit juice formed, it has that dirty looking or broth is there in case of fruit juice which is formed, and the broth can be removed or it can be cleared only by allowing it to rest with pectinase, because pectinase will digest all the outer covering which has pectin, and once it is digested, then there is no problem in the process of or the entire area or vessel in which the fruit juice is kept or vegetable juice is kept. It will get cleared off because there is no pectin left in the juice. Same is about the proteases. They are also used for converting most of the proteins into smaller particles, that is the amino acids. Now in the case of pectinase, which are the different industries where they are used? They all are used in any industry where there will be a raw material containing a fruit, a vegetable, or a wood or plant based product. So textile industry, in case of jute, cotton, anywhere you see, there will be presence of carbohydrate, or there will be production of pectin in the fruit or in the entire plant body. In case of jute, the entire outer structure will be having pectin present within it, the cell wall, so that can be digested with the help of pectinase. Plant ba- plant-based fibers, water, wastewater treatment, paper making, coffee and tea fermentation. Most of the places you can see that there will be presence of pectinase or protease. Uh, Here it is all about pectinase, so there will be a requirement of pectinase, or it can be um, it must be required for for these all industries. While oil extraction industries require the presence of pectinase, again sunflower oil or soybean oil or any vegetable oil, it is made from seeds or it is made from the plants. These plants will be having outer covering of pectin, and that can be only digested with the help of an enzyme pectinase. What about the proteases? Proteases are required in leather, detergent, pharmaceutical, baking, or meat processing industries for production of smaller units of proteins, that is amino acids. In case of dairy industry, it is required to produce cheese or various the proteins are formed in case of dairy industries. Think about detergent. We have already discussed about the micelle formation. Pharmaceutical industries require the presence of proteases. Even baking industry requires the presence of proteases. The reason why we are requiring both these substances is just to make the things or substances available for the easy usage of juices or for production of um fine products like the oil and paper. The next kind of enzyme is streptokinase, and it is obtained from Streptococcus bacterium. It is used in medical for clot busters. What is the meaning of clot busters? In case of myocardial attack, during that time, the Streptococcus is modified in such a way, or it is genetically engineered to form variety which is capable of secreting streptokinase. This streptokinase is a kind of drug which is directly ejected out in the area where the blood clotting has taken place. Most of the time is myocardial infection over where it is used, and during this time the patient requires very quick attention, so that the blood clot has to be removed immediately. So with the help of injection we can just provide the streptococcus to that area. This streptococcus is genetically engineered, so as soon as it receives or it enters a place where there is presence of a blood clot, it will start secreting the streptokinase. It is a kind of thrombolytic drug, and because of that the thrombin will get activated and the entire process of blood clot formation will be ceased. Now, the blood clot formation will be ceased because the thrombin is no longer functional. From prothrombin to thrombin conversion is the primary step for blood clotting. If this step is blocked, then we cannot expect the later on steps to take place. And as a result, the individual who was having a blood clot will now be refrained from the blood clot and will be and that blood clot which was already present can be removed with the help of surgery, or it can be used or certain other techniques can be used for removing it. But in natural way, you can just think that we have to genetically engineer a streptococcus bacterium in such a way so that it is capable of releasing the streptokinase enzyme at the site of blood clotting in the vessels. Mostly it is used in case of myocardial infarction. So these are some of the chemical enzymes which are these are some of the enzymes which are extracted from the microorganisms. There are few more bioactive molecules which are uh, present in our body, and these molecules are required in order to make our life much more easier. Cyclosporin A is one of those, and it is extracted from Trichoderma polysporum. You can see the way it looks. This is the bark of a tree, and over there, this kind of growth, wherever you see, it is Trichoderma. It is a kind of colonizer which has presence of cellulosic materials, and it is present almost everywhere. Wherever there is any decaying surface present, there will be presence of this Trichoderma, and the function is as such that it does not allow any pathogenic attack to take place within the plant body. It can be used as a biofungicide because wherever it is present, it is not allowing any pathogens to enter inside the plant. So the roots will get protected from the pathogenic attack of uh, any other pathogen or any other organism in case of plants, and the plants will be saved from any organism. So it is acting, since Trichoderma polysporum is a fungus, so it is acting as a biofungicide. It is a sustainable development method by which we can fight against any kind of pathogen for the case of various plants. Now, the Trichoderma polysporum is used in pharmaceutical industry for immunosuppress as a immunosuppressive agent during organ transplantation. It means whenever the individuals they are there is a organ donation going to take place, or any individual is going to take a kidney transplantation within themselves. During that time, the problem may arise that the individual they are incapable of transferring, or they're incapable of allowing the organ to fit within their body system. So it is very important that our immune system needs to be seized, or it needs to be inactivated during the time when the transplantation of organ is going to take place, so that no reaction by the HLA complex occurs, or any MHC molecule it takes place. So in that way, immunosuppressive agent cyclosporin A is used as a immunosuppressive agent in case of various organ surgeries or organ transplantation. Next is a statin. It is extracted from yeast, and this yeast is a red colored yeast. Its name is Monascus purpureans. It is used for no, it is extracted from rice. Rice is rice has presence of this red color, or red rice is there which contains that yeast, and from the yeast we extract the statins. The statins are capable of lowering the blood cholesterol level. It also has presence of lovastatin, which is used by various pharmaceutical industries to prepare substances or tablets containing, or tablets uh, which are given to people suffering from obesity. So in that way, cholesterol level can be controlled with the help of statins, and these statins can be extracted from the red rice, which is very rich in vitamin K as well. Not vitamin K, it is monocholine K, and this monocholine K is a active ingredient of lovastatin. Lovastatin is a drug, or is a main ingredient which is used for preparation of pharmaceutical drug against cholesterol. So these are some of the enzymes and bioactive molecules which are used by the pharmaceutical industries, or, and microorganisms are used to extract them